All right, hi everybody. Beepool, I should say. Welcome to BeeCon and my presentation. I'm going to be presenting some of my work from my postdoc with Dr. Nigel Rain at the University of Guelph. We looked at the effect of systemic insecticides on the movement of bumblebee queens using a pretty cool method, if I do say so myself. And it's all fresh off our studio, but you're welcome to share it and look at my awesome TikToks. So why am I using bumblebees? Because they're the best. Full stop. That's the only reason. You don't need any more explanation. Well, because they're also important pollinators of wild plants and of crop species. Of course, we all know that, especially bombus impatiens. And this species can commonly be found in agricultural areas and in our wildlands. As we all know, anytime you go out and survey some flowers for bumblebees, you're going to find impatiens pretty much on the East Coast anyway. And for our method, size matters. We need big bees for our methods, which, spoiler alert, is radio tags. So they need to be big enough to carry the radio tags. And of all of the cast of bumblebees, queens are the largest. So they're the best able to carry the radio tag. They're also really important to know information about their movement because they obviously are the ones that are responsible for reproduction, dispersal of the colony. So their success and their flight patterns really matter for conservation. And what are systemic insecticides? They're chemicals that can travel all throughout the plant. They are systemic. They go through all aspects of the plant. They are very persistent. They're easy to use. They're water soluble in most cases. So they're very popular in the agricultural industry as well. A very common example, or an infamous example, I should say, are neonicotinoids or the neonics, which you've probably heard in the news, are very bad for bees. How can bees be exposed to pesticides if it's supposed to get rid of our pest species and not our good species? There's a lot of ways. You can be directly sprayed on. They can have some drift onto wildflowers that they're foraging on, even if it's not the crop. They can get exposed by the soil. They can get exposed by planter dust. Bees hibernate under the ground and their colonies are underground, so they can get in contact there from the contaminated soil. So there's a lot of ways that bees can be exposed to pesticides. In our experiment, we tested 2 different classes of pesticides, and we did this because imidacloprid, which is a neonicotinoid, its mode of action is on the brain. So to keep this straight in your head, brain icon means neonic, the imidacloprid. And we already know this one's bad. We've got lots of evidence of sublethal effects, including on flight. So this is kind of like our positive control. This is going to be bad for bees. The other one is a newer class of pesticides called cyanotranoprol. It's in the diamide class. It acts on mussels. So that could really impact flight because bees obviously need to use their flight muscles to fly. But we don't know a lot right now about the sublethal effects of this class of pesticides and cyanotranoprol in particular. But it is less toxic than imidacloprid. So the thought is that if we're going to start replacing neonics with something else, and this less toxic one is better, that this would be potentially a better alternative. So our hypothesis then was control, because it's not being exposed to any chemicals, is going to perform the best on whatever flight performance metrics we're looking at. Cyanotranoprol will be kind of in between, and then imidacloprid is just not going to perform very well at all. So our methods, we had bombus impatiens queens, colonies that we raised in the lab until we got the gynes. We plucked the gynes out of the BioBest boxes, and we dosed them with field-realistic concentrations of cyanotranoprol or imidacloprid. We got these field-realistic doses from the PMRA's documents themselves of what honeybees were bringing back to the hive in their nectar. And then to detect the bees, we had to set up some radio towers. By we, I mainly mean me and a couple volunteers who helped get strong and lose us COVID weight and put up 43 radio towers, which is shown, I don't have a cursor, I guess I do here. And all of these little dots here, all the 43 radio towers we set up, and little transparent circles, their rough range that they can detect. And this was done at Rare Charitable Reserve in Southern Ontario. And then we had attached the radio tags. The radio tags weigh about 0.13 grams, which is about the weight of like 2 or 3 crayons in your hand. For size, there's a Sharpie in the bottom left corner. We did trim the antennas a little bit because they're about 10 centimeters long, just so they don't get tangled up in the vegetation. And the method of attachment is very, very, very scientific. It's just super glue. We cool the bees down so we can attach the tag. We attach the tag and away they go. So we measured a bunch of different response variables, including minimum convex polygon, which is a really crude estimate of home range, or like how far the perimeter of their flight range was. Kernel density, which is how much they stayed in that one spot. So the more they were there, the more dense that spot was, which means they liked that spot. Step length, which is a measure of how much they want to stay in that spot. It's from point A to point B. So it's like when you go for a hike and you see a nice flower patch, you want to stay there. So you slow down a little bit so your hiking partner doesn't notice you're looking at the bees. And then when you want to get out of there, like when you didn't hand your paper to your supervisor yet, but they're coming down the hall, you do a larger step length to get away. That's what when you're in a bad spot. And then we looked at different behaviors. So foraging, transit, and resting. Our explanatory variables, we're going to compare our land cover variables as well as time of day, so daytime or nighttime. Here are some little GIFs I made of the flight path of just 3 randomly selected bees of the different treatments, just to show that they did fly and they flew all over the place, and it's pretty cool. So this is just to, I guess, show off a little bit. And I want to test some results. So this is the minimum convex polygon for our 3 treatments, sorry. There's no difference between control and cyanotranoprol, but imidacloprid flew much farther than the rest. This could be because of the excitation properties of neonicotinoids, which isn't that good. We don't really want queens, especially gynes, the ones in later fall, to be flying too far because then they're burning their reserves that they need to overwinter. And then these are the kernel density by land cover. So during the day, there isn't much difference between our 3 treatments. They look pretty similar. They're using the same sort of land covers, and they're most often in flowers. But at night, there's a big difference. So something's going on to impact their behavior, especially at night between the treatments. And here are the step lengths by land cover results. So there isn't much difference. They're doing what is expected, which is good. They have lower step lengths, so they like the flowers. The biggest difference is in some of the more developed land covers, and it's not even that big of a difference, but for some reason, cyanotranoprol and imidacloprid are thinking that crop is a good thing, and the crop was mainly corn or soybeans. It's not the best for bumblebees. The biggest difference we noticed was in behaviors, a huge difference. Control bees are just lazy. They want to rest all the time. And then foraging sometimes, which makes sense. They got to build up their fat reserves to survive hibernation. But imidacloprid and cyanotranoprol are in foraging behavior for a lot of the time. So that's really impacting their behavior. So my result summary, the largest difference between our treatments was for behavior. We didn't see much difference between flights. Some of the not presented results was total flight distance flown for their whole flight path. There was no difference there. But the biggest difference definitely was their behavior and the response to land cover for some reason. So just to rehash my hypothesis, this is what I thought was going to happen. This is what I kind of hoped would happen, but it's more like this. So cyanotranoprol and imidacloprid are having basically the same impact on bumblebee queens, which is not a good news story. It means cyanotranoprol is not a good replacement for imidacloprid. We should not be using it based on my results so far. That's kind of what I'm getting at with my research impacts. So as neonics are becoming increasingly regulated, especially in the European Union, and they're slowly being phased out, we're looking for different pesticides that can replace them. And cyanotranoprol and diamides are kind of suggested as a potential replacement. So it seems like they're going to have similar or the same sublethal effects, so they're not going to be the greatest replacement. So next step, I'm going to look at, so I was just in the field even last week, so these are very fresh results. I'm going to start looking at the duration of the flight periods, the number of days they were flying, and see if that's different between the treatments. I want to look at the characteristics of the last detection point to see, with the assumption the last detection point is where they potentially overwintered, to see if we can get any characteristics of what that overwintering area is like. Then there's some wild gyne work, so we tagged wild gynes as well. We have our spring projects where we did very similar experiments. And then because these results are so fresh, if there's any other ideas or feedback from the audience, I would love to hear it. And with that, I would like to thank everyone that helped me, especially putting up the towers. WPC made a very big appearance. Thank all of them for coming out and volunteering, as well with the bumblebee tagging, because that was a huge endeavor. And I will take any questions. I'm gonna clap for myself.
Questions, I just need technical stuff, so just ask questions.
Sure.
We do have time for questions, so go ahead.
Yeah.
I'm wondering, is there any work in your lab or other labs, is there similar effects on the reproduction?
For the diamides, I don't, so it would be Sabrina's work would be the closest, and I don't remember what her results were for the bumblebees. So I think there was, and I actually don't remember if she used diamides on the bumblebees. Yeah.
So it'd be interesting to then meet people. And I guess I have a follow-up question. If like the foraging, the radio, that's a really interesting setup. I was wondering how often do you lose a bee in that? Like, do the bees go out of range very often, or do they disappear entirely? I guess kind of like how many bees do you start with and how many are useful and then?
So we started with 75. We had 25 for each treatment. Most of them were in the study area and detected. There was 5 in total that, like, once we tagged, just never came back. But there's a few that were in the study area, so we saw them and then we saw them leave and then never come back. Or we have ones that are doing weird stuff, like they're coming in and leaving, coming in and leaving, because we can't tell what they're doing once they leave the study area. But a lot of them stayed, which was great. It's like some of them we have thousands of data points for that one bee.
Or the nest central then, or?
Well, these were the gines in the fall. So we just took them out of the BioBest colony and then released them at the site. So from the release site, it's also kind of cool. Most of them stayed near the release site at first, and then they started doing big exploratory flights around the site. And then you can see them like narrowing in on this one area that I think where they overwintered, which is pretty cool. Not necessarily more activity. They were moving at night, which I was very surprised about. All treatments, not even so neonics have been shown to make them active at night when they're not supposed to be, but all treatments were moving around at night. Not really sure why, but I was surprised by that. Yeah. Sorry? Could be, yeah. The site is kind of remote, but like Cambridge is right next door. So yeah. Oh, I didn't check online.
Oh, and I have a question, if I may, actually a comment.
Sure.
So neonicotinoids are thought to primarily work on kind of the brain and the nervous system. However, in insects, they continue acetylcholine receptors, so the actual receptors neonicotinoids target, those seem to be present everywhere, not just in the brain.
So I have to update my icon then.
Yeah.
Has to be the whole bee. So there was a couple of questions. Do I have time or no?
Yes.
Okay. Do you think that learning measured by proboscis extinction responses, PR, would differ in a similar way? That's for me. Oh, this is for me?
Okay.
Yes. I think so. I think it would be different. I think maybe we'd see a stronger impact with imidacloprid versus cyanotroniprol, just because it's not supposed, apparently it's not impacting the brain. So for the PER experiment, I think we would see a more negative impact of imidacloprid versus the control or cyanotroniprol, but I'm totally guessing, I'm not sure. And then Tiffany, do you think those late data points could help inform site selection for overwintering? Potentially. I mean, there's a lot of caveats with that because it's just the last point of detection. So if I do analyze those results, there's gonna be a lot of asterisks beside that. It's the last point of detection. It looks like they're all disappearing at the same spot, and it's kind of like right in the middle of the study site. So it's unlikely that it's something like they flew away and they weren't detected anymore. So I think it's an overwintering site, but still we can't say that. Like maybe there was something sitting there and eating all my bees at that one spot. The tag all fell off. They all died there for some reason. Like I can't know for sure. So like there's lots of asterisks beside there, but it's also like really hinting that that's a good overwintering spot. And it's a north facing slope, but it's not treed or anything. It's an open field. So it kind of goes along with what we already know about overwintering.
All right, I think we're gonna wrap up
Yep.
this.
Yep.